Okay, hello everyone. Welcome to this third day of the CompuSol 3D workshop. Today I will be presenting the module 3.1, introducing cell properties and how to tie some of these properties to cell fields, to chemical fields. Just as a reminder, this presentation will be recorded and live streamed. Again, I highly recommend everyone to download the demos and upload them to NanoHub if that's a platform that you're using, and upzip them locally if you're using computers on your local machines. So as I mentioned, we'll both be talking today about cell properties, in particular cell division, how to implement cell division, cell growth, and cell division in CompuSol, and how to tie some of these properties to the behaviors of the chemical field. In the first part of the module, I will be talking about how we can think of cell proliferation from a modeling perspective, how we implement it in CompuSol, and what are the considerations that we have to take when we are making our cells grow and divide. So the basic principle of life really is that cells have to undergo proliferation and division. And in cellular organisms, it's particularly important for cells to decide when to proliferate, when to survive, and when to die. And these decisions are made based on signals that the cells receive both internally and externally. And there are factors such as cell contact or factors such as cell-to-cell contact that might inhibit this cell growth and proliferation, and other factors such as growth molecules that might induce cells to proliferate. And we will see examples of modeling these two types of behavior. From a biological standpoint, when cells undergo proliferation, they usually start by doubling the DNA material in the cell. And once they have doubled this in this S phase, when they have doubled their genetic material, they undergo actual cell division. And then after they remain in a rest state where they decide, or they cross certain checkpoints where they decide whether they want to continue proliferating or they want to arrest cell division. And there are multiple of these checkpoints along the different stages of the cell cycle. I'm not going to go into the details of these because from a CompuSol perspective, we are not going to model the cell cycle in detail yet. And I said yet because tomorrow we will see how we can couple the cell division that we will be discussing today with internal models of the cell cycle, with internal models of the cell cycle that might control how the cell divides. But at least for the purpose of this module, the basic idea behind cell division is when we implement it in CompuSol is that we will make our cells grow, and they will divide when they reach a particular size. And I'm sorry that my screen is a little bit small. I am It's because I have this split screen, so because I want to be able to share my code and my presentation. So, but the slides are also shared on the Google Drive. If you want to have it as a reference, so you can see it side by side while I'm talking, you can also download them. Juliana, if you can post a link to the directory with the slide decks. So coming back to the idea or summarizing here, the factor and processes leading to cell division and proliferation are very complicated in the real world biology. For the purpose of this module, we will simplify all of that complexity by modeling cell division and so I'm sorry. We will simplify cell division by having our modeling as our cells growing and as our cells dividing once they reach a particular size. So how do we make our cells grow? So in CompuCell, the simplest way we can make our cells grow is to increase our the target volume of the cell by a certain amount each time step. How we implement this cell growth can be as complicated as we want, and we will see different ways in which we can modulate cell growth. But again, the idea is that always the idea will be that we will increase the target volume of the cell, which represents a increase in the mass of the actual cell. And then once we have reached a particular size, then we will have that cell divide. Another important thing to notice here is that we actually do not increase the actual volume of the cell. What we increase is the target volume, and the actual volume will be the result of the interaction between the different energy or constraint terms that we include in our simulation. So we can set ourselves to, we can set the target volume or increase the target volume constantly, but the actual cell volume of the cell, whether it reaches that target volume or not, will depend on other factors. So for example, whether the cell has space or not to do it, or whether we adapt what the surface area of the cell should be. And all of those factors will play together to determine what the actual volume of the cell is. But in principle, the best way we have to control whether we want the cell to grow is to increase its target volume. Now, and remember that increasing target volume will the target volume acts as a spring force in our energy, in our energy terms, that basically forces the cell to increase. Another thing that is important to notice is that we cannot define, so far we have been doing a lot of modeling from the XML, but if we want to be able to increase the target volume of our cells, we have to do it dynamically from the Python script, and we will have to define it inside the Python script, and we'll have to disable any specification of the target volume in the XML. Otherwise, we wouldn't be able to. So the first exercise, this is exercise 311 in the demos that I shared. What we will do is that we will create a simulation domain, we will have a single cell, and we will assign contact energies, and we will assign the volume constraint. And we will show how to select a growth steppable from the wizard, so that facilitates us implementing cell growth later on. Then we will determine, we will initialize a single cell in the center of the simulation domain, and then we will have this cell growth by setting a growth rate of 25/500. And then we will, after having also growth just with the volume constraint, we'll add a surface constraint and see how the surface constraints affects the cell growth. Okay, so first, to start the simulation, let's start creating the simulation. So we create a new project. I'm gonna call it growth here. And then, I'm sorry. And then I'm gonna create a 100 by 100 lattice. Now I do have cells that move, so I'm gonna set my boundaries to be periodic. I don't want the cell to bounce against the borders of the simulation. We talked about what periodic boundary conditions meant yesterday. If anyone wants a reminder, I would direct them to the presentations yesterday, but the same ideas that apply to the field apply here to the simulation. I'm gonna use the Blob Initializer. I'm gonna create one cell type called cell. And I'm not gonna add any chemical fields. And now I'm gonna add the contact energy constraint, a growth steppable, and the volume local flex. Oh yeah, I'm sorry. So I did mean to change it, the pixel copy range to 3. Thank you so much for the reminder. Yep. Okay. And once I've selected all of the properties that I want to add, then I can finish my, I can finish with the, I can generate the simulation. So once I've generated the simulation, the first thing I should notice, yes, please. I forgot to mention it. If you didn't, you can mark the surface local flex. If you didn't, I'll show you how you can add it later on, because if you forget to add some of the properties, you can always use the pull down menus in Intuitive to add the steppables or the plugins that you might need to add. So if I go to the XML on this file, I would notice that I've defined my simulation domain. One second. And I have 2 cell types. And notice that I now have this surface and volume plugins, and that they do not have any information. Now, let's say that we forgot to add the surface plugin. I can always come to CC3D ML, plugins, and then I hit and surface. Right? and then it will paste a snippet with the information to specify the surface plugin. However, we want to be able to specify the parameters of the surface constraint in the Python's steppable. So we will have, we want to delete, get rid, or comment out the information inside the plugin in the XML file. And then we will also notice that I have now the contact energies. We're not gonna play with these contact energies now. And then that I have a blob initializer, and I've set the blob initializer so that I initialize one single cell in the center of the simulation domain. So I just set the radius to 5 and the width of the cell to 5. Okay, now I said that we have loaded the surface and the volume plugins, but we haven't specified what the properties of these constraints or the parameters of these constraints are, because we will do it in the Python step. So if you don't have the blob initializer, you can again always come to CC3D ML steppables, and then then you can delete the information in the blob initializer, come to CC3D ML steppables, and then you can click on blob initializer, and that's gonna add this code snippet here, and then you can modify it as in the yellow lines in the okay. So now if we come to the step, as I was mentioning, if we come to the steppable, we will see that that we have specified that we now have to specify what the cell target volume and the lambda volume is. And then we will see that we also have this growth steppable that allow us to increase what the target volume of the cell is. So all I need to do for this particular simulation is to set the target volume, and since this is happening at the start function, this is gonna be the initial target volume. So I just have to set that one to 25. I need to set my lambda volume to 3. And then in the growth steppable, I will increase my target volume by the rate 25/100, which means that every 100 Monte Carlos, the cell is gonna grow 25 pixels every 100 Monte Carlo steps. Okay. So I'm gonna wait for everyone to catch up here before running the simulation. Okay, good. It looks like everyone is with me. So now if we execute this simulation, what we see is that as the simulation times goes on, my cell increases because I am increasing my target volume. Okay. So I'm gonna stop this simulation here now. and continue talking about how I could add more constraints to this simulation. I'm sorry, there's a couple of questions. Let me come back to that. It's not, right. It doesn't happen every 100 steps, right? It continuously, so it basically increases 0.25, the target volume, every Monte Carlo step. So I can show the XML again, but I didn't do anything fancy. I just initialized a blob initializer with a radius 5 and a width 5. That's gonna give me a single cell in this center of the simulation. Radius means the radius of the blob, as people are discussing, not the radius of the cell. Width is the length of the cell. Okay, so now we can add an additional, because we have the in the XML we also have the surface constraint or the surface plugin loaded. We can also specify an additional constraint in our cells, and that will be a target surface. And by surface, we mean the number of pixels that are the boundary of the cell. And again, since I've commented out that information in the XML because I want to be able to modify it on the fly. On the fly, I mean, as at a constant rate, I can specify it here in my steppable just below where I've specified the target volume and lambda volume. And it will be target surface and lambda surface. Okay. So I'm just gonna do that now. So I'm basically adding a target volume, a lambda volume, a target surface, and a lambda surface. Oh yeah, there is a typo in the slide. I'm sorry. It should be lambda surface on the last line here. I'm sorry about that. Thank you for catching that up. So quick question, what do you think it's gonna happen once I run this simulation now, right? Because my target volume, it's increasing, but the target surface is not, right? I'm not readapting, right? So the surface area will constrain what the volume increase will be. So let's run, now that we have, let me make sure that everyone is with me. So just as a recap, I first initialized the volume constraint, and I am increasing the target volume by a particular rate every Monte Carlo step. And now I am adding an additional constraint, which is the surface constraint. And now I can execute my simulation again. And as Some people mentioned in the chat the surface constraint is now constraining the volume. The cell is very squared because I'm basically adding more and more target. I'm increasing my target volume, but I'm not readjusting my target surface. So it basically wants to increase. It wants to increase the target volume, but the target surface remains the same. Good question. Why is it still increasing even if I am not increasing the surface area? Well, because or the target surface, and the reason it's because there is always a interplay between the different constraints. It's not like one constraint totally inhibits the other, right? So what's happening is that the strength of these constraints are similar, and the magnitude of these constraints are similar. So the cell is still trying to achieve or meet the both of the constraints at the same time. So it still increases the area, but it's not. Let's say it still increases the area, but it has what we'll see later. We'll call later. It's under a lot of pressure. We'll come to that idea later, and we'll see how that. And that's why the cell it looks very square and not very motile in this particular simulation. But the idea here is basically that we are putting too much air into a balloon or too much water into a balloon that is relatively small and it's very, very tight. It's very under a lot of pressure, but that prevents the cell from moving. If I wanted to, for example, if I increase the lambda surface by a big factor, so I make the importance of the lambda surface much bigger than that of the, I'm sorry, if I make the importance of the surface much bigger than that of the volume, then in that case, I think it was Polly who had that question. Yeah, it's not growing anymore because now the surface constraint is much important than the volume constraint. So the cell will try to meet the surface constraint rather than the volume constraint. Okay. So let's put back this to 3. So we're basically making them the same, about the same magnitude. So the way we can solve this, if we have these two constraints, if they have the interplay between these two constraints, is to adjust the target surface of my cell as I increase the target volume. So yeah, that's what you see. So the way I can adjust the surface as a function of my target volume, there's many ways you can do this, but the easiest way is to assume, for example, that my cells should occupy the area of a circle. And then given the new target volume, I can infer what the target surface would be from that target volume. I think it's divided by 2 here, but just as an approximation. And then I can readjust my target volume, my target surface. I'm sorry, this doesn't work here. I can readjust my target surface every time I increase my target volume, such that the target surface readjusts itself So I'm gonna relaunch the poll just to make sure. I'm sorry. I'm sorry if I take a little bit of time. I'm just trying to keep up with the chat to see which questions I can answer and which I cannot. So the reason it's pi in this particular simulation is because I've already, and I forgot to mention this, thank you for the question. I've imported pi from the math library. So it's available to me. And let me relaunch my code. Let me come back to and relaunch my code. So all I did was, after adding my targets, my surface constraint, I'm modifying the surface the same way as I adjust the target volume. Okay, good. Everyone is with me. So now when I execute this simulation, sorry about that, I notched. So the cell starts growing too, and I said that I have a factor of 2 missing somewhere. That's why it looks a little bit squidgy in the edges, and that's because I have my target surface is just a bit bigger than my target volume. Okay, but now I can meet both constraints at the same time, or my cell can meet both constraints at the same time, because as the cell increase, as the cell volume increases, I readjust what the target surface should be. Or as the volume increases, I readjust what the target surface should be. Yes, AJ, I'm sorry. For those to import Pi, there's many ways, many libraries you can import it from, but probably the easiest one is, sorry. From math import Pi. Okay. So I have this version of the code in exercise 311. Some of the additions are commented out in the original version, but you can play with the different combinations of the constraints and how to modify them. So now that we have make our cells grow, now we can start thinking about how to trigger cell division. It's really, there is no predefined condition to in CompuCell to trigger cell division. It's up to the user to define what condition will trigger the cell division. As I mentioned at the beginning, in real life, there's many conditions cells have to meet in order to be able to divide. They have to have the right signals. They have to have enough nutrients. They have to have enough space. They have to double their genetic material, and then they have to undergo mitosis. In CompuCell, as James mentioned at the very beginning, this is nothing happens unless nothing exists, and nothing happens unless I explicitly state it. So I have to state what the conditions are for our cells to divide, and I have to be I have to make sure that the cells are able to meet those conditions if I want them to divide. And once they've met those conditions, then there's other options or other factors that I have to take into consideration in terms of what happens during and after division. So what plane the cell's gonna divide, I have to be able to specify that, and whether after division the cell both daughter cells have the same side, or there's a cell that is bigger than the other. Normally, we think of cells as dividing into two of the symmetric cell division, where cells divide into two cells of the same size, but that is not always the case, especially during development. So again, the simplest condition that we can think of, or that or we can implement in CompuCell, is to have our cells divide once they have double their initial volume. And once they have divided, then we have to have the daughter cells inherit modified versions of the target volume and the target surface of the mother cell, right? If you imagine that the target volume represents the mass of the cell, then you should expect in symmetric division, you should expect the daughter cells to inherit half and half of the volume of the volume of the mother cell. When implementing cell division in CompuCell, there is this special steppable called Mitosis steppable that has a little bit of a different structure and has it has all the available options that you will need to implement cell division. In particular, it will have it will ask you to specify what the condition is and what happens to the cells after they divide. And just as a teaser here, the Mitosis steppable has this structure where in the step function we have this cells to divide dictionary, and once we specify what the condition for cell division is, we append the cells that we want to divide to this, it's not a dictionary, I'm sorry, it's a list. We append the cells to that list, and then we iterate through this list and perform cell division on those cells. And then we can divide them along random axis, or we can actually specify what axis we want the division to occur. And then we have this special function called update attributes, where we decide what happens to the attributes of the cells once they divide. And the way to specify, the easiest way or the simplest way to specify is to clone the pattern properties to the child, but we want to divide the target volume of the cell in half, or the target volumes of the daughter cells in half, such that the mass of, such that we divide the mass of the mother cell into the daughter cells. And there is more control options that we have, but we won't go into those right now. We'll see those in later in the exercise. Okay, so let's go ahead and start. Let's I'm sorry, I deleted one slide that I wasn't meaning to, but what we will do is that we will create for this exercise, we'll create a simulation domain, and we will add two different cell types, stem and terminally differentiated. And we will add constant volume, growth, and division from the cell properties. And we will initialize a single stem cell in the center of the simulation domain. So let's do that together. new project. I'm gonna call it division. Again, 100 by 100. And I keep doing 100 by 100, but again, this is up to you whether you want more or less resolution and more or less space. We're gonna have periodic conditions on the X and Y axis, the Blob Initializer. We will add the stem cells and the terminally differentiated cells. No chemical fields. And then we will choose, again, pixel copy range 3. It's always good to avoid lattice effects. And then from the sub properties and behaviors, we're gonna choose contact, growth, mitosis, and volume local flex. I'm gonna wait for everyone here. Just 1 second. So you can specify it if you're interested in adding a surface constraint. I'm not gonna be, just for simplicity, I'm not gonna be adding the surface constraint again in the exercise of this module. But if you want to add that surface constraint for any particular reason, depending on the model of interest, you could also add it. But we won't for the purpose of this module. Okay, and then we hit next, and we finish, and we open the simulation. What I'll do is I'll open instead of typing in everything from scratch. I'm just gonna open the exercise 312, which has the code for this exercise. Okay. And then we can see that we have created 2 cell types. We have the contact energies between them. We're initializing a single stem cell in the center of the simulation domain in the Blob Initializer. So if you're typing this from scratch, make sure you adjust that. So I always use a radius of 5 and a width of 5 to ensure that I have 1 single cell in the center of the simulation domain. And that my boundaries are periodic. Okay. So the reason I have 1 type of cell is because I'm gonna start with 1 single cell, and I want it to be of the stem cells for the later on the exercises. I want to start with the stem cell for later on the exercise, so I just went ahead and set it here. If I said stem and terminally differentiated, it will randomly choose which one of them to start with. But I always wanted to start with the stem cell, so I just set the type of the cell I'm initializing to be a stem cell. Now, if we come to the steppable files, there's more stuff that I added here. But if you start from scratch, what you'll have is a constraint initializer with the target volume and lambda volume. You will have a growth steppable. I'm sorry, this is moving way too fast. Where the target volume of the cell is being updated. And you'll have the mitosis steppable that we talked about earlier that has the cells to divide list, the condition for when that division occurs, the the implementation of the division along a random orientation, and then the updates attributes, and then the updates attributes of a special function. Okay. Now, if I run this simulation, so note here in this part of the code that I am actually changing the type of one of my daughter cells. So if the parent cell, let's say, is stem cell, then the daughter cell will be, one of the daughter cells will be stem cell, and one of the daughter cells will be terminally differentiated. So if we run that simulation now, let me make sure that everyone with me before I run the simulation. So again, here, this part of the code is determining that one of the daughter cells is gonna be of type 1, which is my stem cell, and one of the daughter cells is gonna be of type 2, which is the terminally differentiated. Okay, seems that most people are with me, so I'm gonna go ahead and open that simulation in Player. I'm not gonna use this now. So as I mentioned, we start with a single cell in the center of the simulation domain. So this, Polly, you said this part is missing for you. It's fine for now because we won't do anything with it right now. But we'll come back to that a little bit later during the exercise, and it's not that complicated, so you can type it directly if you're missing it. But now for this part of the exercise, okay, it crashed. 1 second. I'm gonna have to relaunch Compusol here. Did it crash for anyone else when they tried running it? That's exercise 312. Okay, it ran for you. Okay. Okay, I'm gonna minimize this. So what I can see is that my cells are growing and they are dividing, and then I have cells of both types. In this case, blue is the stem cell and green is the terminally differentiated. And the reason I have cells of both types is because when my cells grow and divide, one of the daughter cells is going to be of one of the types and one of the other one is going to be of the other. So should have about the same number of cells for each type in the simulation. I'm going to start it over. And if I let it run for long enough, the cells are just going to fill up the whole simulation domain, and they'll stop growing and dividing because they have no additional space to do so, and we'll come back to this idea later. Okay, so yeah, Ying, we'll come to that idea later. that's why it's added. That's why the plot is added in the code, because we'll discuss what's happening. So now we can try different conditions. We can try dividing it. I'm gonna leave this to you, but you can just change when what the condition is, and you can tell. So for example, instead of doubling when it reaches a target volume of 50, we can make them divide when they reach a target volume of 30. Or we can also divide them along different orientations. Okay, but I will leave this part of the exercise for you to do as a take home, so you can explore what the effects of these of changing the conditions are. So now, so how to control the rate of division? So the rate of division I can control by changing what the rate of growth is, right? Because that's the only condition that we are implementing for our cells to undergo cell division. And I do so in the in the part when I'm modifying the growth rate, I can make it grow slower. Sorry. So in the part when I'm making our cells grow, instead of 25/100, if I do it 25/500, then that will control how fast cells are growing and consequently how fast they are dividing. Okay. Now, I want to, instead of having all of our cells grow and divide, I just want my stem cell type to grow and divide. And once they are in the terminally differentiated cell, I want them to not grow and not divide anymore. So again, there's multiple ways I could implement this, but the simplest way is to modify which cells I'm actually increasing the target volume. So if we come to the code in line 44 here, in line 43, you see that I'm iterating over in the growth steppable, I'm iterating inside over all of my cells, which is my cell list, and I'm increasing the target volume of all of my cells. But if instead of doing that, I iterate over my stem cells and increase the target volume, then the stem cells are the only cells that are gonna grow and the only cells that are gonna divide. And to do that, I have this syntax here for the cell list by type that I can iterate over. And if I iterate only over the stem cells, then I can just increase the target volume of the stem cells. So I'm just gonna comment out this part of the code and uncomment this part of the code and launch my poll. Just to make sure that everyone is with me. Okay, everything I did here was there's some people that are still working on it. So the only thing I did, right, for the purpose of this part of the exercise, all I want to do is I want to have the stem cells grow and divide. And the way I do it is by, instead of in the growth step, instead of iterating over my whole set list, I iterate over the set list by type. And the type I've iterated over is the stem cell type. And see how I reference the type is by referencing self.st, which is my stem cell. And those are the only cells that are gonna grow, and consequently are the only cells that are gonna be able to divide. So let's run the simulation again. So now you can see that only my stem cell is doubling in size and dividing. Once it's divided into a terminally differentiated cell, that cell doesn't increase in size, so it doesn't divide. So now I've shown you how you can have better control over right. I mean, as I mentioned, Polly, there's multiple ways you can try to implement this. I chose to do it by just having the stem cell divide. But I could also have, for example, all of the cell growth and only my stem cell divide. I could do so because in the mitosis steppable, I also iterate over my whole cell list. If I were to iterate over the cell list by type, then only those cells will be appended to the list that I'm using to divide cells. I don't know if that makes sense to you, but right. So the condition, it will change, and again, but then what will happen in this particular case is that all of my cells will grow, but only my stem cell will be the one dividing. And you can try that yourself as a homework too, if you want to. Okay, I'm gonna continue moving on because there's other things I would like to cover. So as some people notice, this code already had a, and that's because I forgot to delete that floating code from this exercise. But this exercise already had a code where I was plotting the average volume of the cells in my simulation. And what some people noticed was that as when the cells occupy the full space of the simulation domain, that the average target volume will continue to increase. And the reason that it's happening is because that we have no way of, we haven't specified any way of the constant, the growth rate is always constant, right? And the only way that the target volume decreases is when the cells divide. But if the cell cannot divide because it doesn't have space, then the target volume will just continue to increase. That's what we call the cell. That's when we call our cells to be under a lot of pressure. And pressure is just the difference between what the target volume of my cell is and the actual volume that the cell wants, that the cell has. So if the target volume is much bigger than the actual volume, then we say that our cells are under high positive pressure. Okay, and there is a derivation that we can do from the energy term to the pressure. But in particular, but we won't get into the details of that. What is important to notice for the purpose of the module is that the pressure of our cells is proportional to the difference between the target volume and the actual volume. And if there's no more space for our cells to grow, they won't divide, and the target volume will continue to increase, and so will do the pressure, or our cells will be under a lot of pressure. If we wanted to avoid putting our cells into a lot of pressure, we could modify our growth rate such that instead of being a constant growth rate, the growth rate is a function of the pressure cells are under, and there is multiple functional forms that this modification could have, depending on how smooth you want your transition to be between, let's say, cells, between when the target volume stops increasing. So now we will, the advantage of using this modification of the growth rate due to the pressure is that it allows us to implicitly model contact-inhibited growth, right? In the context of cells that are growing and fill up a full space, if they don't have space, to divide into, they stop growing, right? And cells live happy in those conditions. They're not inflated balloons. So we will want to, if we want to mimic that, we want to reduce the pressure our cells are when they occupy the full simulation domain. So we will do that by modifying the growth rate using the pressure of the cell as a reference. I'm going to walk through these. So the first part of the exercise is basically using the same basic simulation specifications as in the previous exercise, but we will add additional information into that one. So I'm not going to create a full simulation from scratch. Well, what I'll do is that first I will add a plot that shows the average volume of our cells. And just as a quick reminder, if I want to add this is inside this 4-cell loop, so it's creating a lot of, let me move this out and move this out. So, so just as a quick reminder, if we want to create a plot window, we go to a start function. It can be whatever start function we want. And then we go to CC3D Python scientific plots setup. And that's going to give us a code snippet that we can modify to include, I'm sorry, here it moves really fast and no, when I scroll, that we can modify for the specific details of the plot that we want to add. Let me decrease a little bit. So we're gonna create a plot window that has as a title average target volume, and we're gonna create a data series called AVOL. And I'm gonna wait for people to do that. I'm gonna give people a couple of minutes to be able to do that. Please make sure that the indentation is correct and that the plot window is not being created inside the cell loop. I'm gonna relaunch to give everyone some time to create the plot window. So basically all we did was created a plot window by going to CC3D Python, scientific plots, and setup. And once we had the cut snippet, we changed the title to average target volume, the y-axis title to volume, and then in the data series, I'm calling the data series AVOL, and that will create the plot window. Next, we want to add data to that plot. And to add data to that plot, I have to calculate what the average volume of in my simulation domain is. And that's what I do in inside the step function in this code. I basically create a variable called avol. I measure how many cells I have, and the way I measure how many cells I have is by measuring the length of my cell list. And then I iterate over my cell list and add the target volume of each of my cells divided by the length of the cell list. And I added into this avol variable. And then I plot the avol variable. And again, just as a refresher from yesterday, if I want to add data points, I go to CC3D Python, scientific plots, add data point. And then I modify it such that the series, the name of the data series is the same that I specified in line 24. And that I'm plotting the right variable, which is in this case is the average volume that I call avol. Okay, I'm gonna end poll relaunch to make sure that everyone is with me. Okay, some people need a little bit of time to work on this. I'm gonna give it a couple of minutes. So just as a recap here, we've basically used the same simulation as before, as the previous exercise. We've just added a plot window in the start function to plot the average target volume. And we have calculated the target volume inside the step function, right? And I've plotted that, I added the data point to the plot that I created. So I'm gonna give people 1 or 2 minutes to catch up here. Also, as a reference, this code is available in the demo 313 or demo exercise 313. So now if I execute the simulation, what I see is that my average volume increases until my cells divide, increases until my cell divides, increases until my cell divides. And if it keeps going until it fills, until my cell fills up the whole simulation domain, and then it continues to increases, right? Because what I'm plotting is that target volume, and the target volume continues to increase. Now I'm gonna add a, what I'll do is I'll add an additional property to the cell dictionary that I'm gonna call pressure, and to add into that dictionary in the start function where I specified the constraints, I also define this entry or attribute in the cell dictionary that I'm calling pressure. I'm also creating an additional second window to plot the average pressure. And then I'm calculating in the step function, I'm basically reusing the code that I had already there, but to calculate the cell pressure instead of the average volume to calculate the average pressure. So in this part of the code, what I do is that I iterate over the cell list, and then I define the cell, the pressure on the cell dictionary to be this formula, which is a formula for pressure that we introduced earlier. And then I calculate and plot the average pressure of my cell. So this code is doing two things, right? It's first calculating what the pressure is that each individual cell is in. And secondly, it calculates what the average pressure in my simulation domain is. So what I'll do is I'm gonna open this exercise, this demo 313. And now we can see the full code there. So I'm defining this this entry in the cell dictionary called pressure. I'm creating a window to plot the average pressure. And then I am calculating both the average volume and the average pressure in my simulation domain, and I'm plotting them in their respective windows. Please do notice that when I create two different windows, I have to name them differently. So the first window, it's named self.plot_win. The second window is called self.plot_win2. And then accordingly, every data series and data point has to be referencing the right plot window that I want to add that data to. And then if I run that simulation, now I have a average pressure and average target volume. And well, in this particular version of the code, I already modify it. But if I don't modify it, what I should see is that the average pressure and the average volume, well, the average target volume will increase, and so will do the average pressure. So now I can modify the growth rate based on this property that I'm calling pressure. And possibly the simplest way of doing is to do it linearly from a reference pressure value. And the reason I'm using max is because I don't want my target volume to decrease. So if the pressure is over 70, which I shows basically by looking at these plots, then my cell target volume will be, the increase in my target volume will be 0. And that's the way I can control whether my cell whether the target volume continues to increase after the pressure is above some threshold, right? So in this case, the threshold is 70, and if the pressure is above 70, then the target volume stops increasing. And that's what I see if I run this simulation. So now the cells do fill the simulation domain, but the target volume stops increasing after they have filled up the simulation domain. And I'm gonna relaunch the poll just to make sure that everyone has been able at least to run this code, because I've uploaded it, and at least that you are that mark yes is you're being able to run the code, and at least you understand what the code is intending to do. Yes, I can definitely show the code again. So here is a part of the code where I modified the growth rate. Let me decrease this a little bit to make it a function of the pressure the cell is experiencing. So 70 was just a number I chose randomly. I chose that from the previous results. I saw that before the cells fill up the plate, they never reach 70 here as the pressure. So I wanted to keep the pressure. I set that threshold that way by looking, but I mean, that number could be way better or less informed depending on the motivation for it. But I just did it by looking at that plot. Okay, so I still have 10 minutes. There's a couple of stuff that I still want to cover. So the next, what I'll do is the next exercise, I'll walk you through it, and I'll show you the code, but Juliano is gonna be talking about it more in detail later. So I'm just, instead of walking through building the whole simulation, I'll just explain the code and explain what it's doing because it's important for what Juliano is going to be teaching later today. And then we will move on to the basis of chemotaxis. So far, we've modified our cell growth using contact inhibition, right? By somehow modeling contact inhibition using this cell pressure or property that we've defined. There's other mechanisms that control cell growth, and that is not just contact inhibition. Or availability resources is also an important mechanism that controls cell growth, and this is something that becomes very obvious in tumor spheroids, where you have nutrients diffusing in from the boundaries or surface of the spheroid, and you can see that at the rim of the sphere, cells do proliferate and grow and proliferate. In the interior, cells die because they are because of lack of nutrients, and there is like intermediate regions where cells are in acquiescent cell where they do not grow, but they where their cell cycle is arrested, and this is controlled by the diffusion of the nutrients from the exterior or from the surface of the spheroid to the inside. And we can model that in CompuCell by coupling cell growth with uptake. And this builds on the ideas that we were discussing yesterday, where we created a chemical field and we had our cells uptake from that field. Now we can tie that uptake to cell growth and control cell division that way. And the idea is simple, and actually the code to implement that is not that complicated. All we need to do is to modify our cell growth by the amount that the cell is taking from the environment. So if the cell takes nothing, then the cell does not grow. In the simplest case, the cell doesn't grow. If the cell is taking as much as it can, then the cell grows as fast as it can. Additionally, there's nothing that prevents us from combining different control mechanisms for cell growth. So we could potentially combine pressure-dependent growth with what I'm calling here diffusion-limited growth. The syntax is quite easily. We're gonna use this uptake inside cell function that we saw yesterday, but we will also add this total count that allows us to recover how much and reference how much the cell is uptaking from the environment. And again, the idea in this code is that I'm going to walk you through it. We're not going to build it together, but we create a simulation domain just as in previous exercises. And then we add a nutrient chemical field. And then we've had our medium secrete this field at a constant concentration that we set to be 1. And again, for those of you who were not able to join us yesterday, you can there's videos of those presentations where we discuss how to create chemical fields and how to set the secretion options. And then what we will do next is that we take from that field, and then we record how much we're taking from that field, and we create an additional visualization field that allows us to see visually how much cells are taking from the environment. And finally, we modified the growth rate by how much cells are taking. So to see this code, we can come to, we can open the demo 314. And in the XML, for those of you who were with us yesterday, you will see it's just the same as the previous simulations, except that now we have added this chemical field by specifying the diffusion solver. I'm sorry, my computer. Do you guys still hear me?
Yes.
Okay. I think it's my nano hub. My nano hub is acting up. So I'm gonna have to terminate it and run it back. But coming back to this, we've just added a chemical field, and I'm not gonna go into the details of this because this was discussed yesterday. Oh, it's my email. And then with just one second, let me make sure that my internet is working. Good. Okay. I'm waiting on NanoHub to load again. But the idea in this code is that we've created a I'm sorry, I'm relaunching the end of module questionnaire. So we are creating a chemical field, and we're having our medium secrete. And then we've also enabled the plugin steppable, the secretion plugin in the XML. And in the step function of in one of the step functions, we have make ourselves take up from their environment, but and ran the simulation. And in this point, what you will see is that the cells grow, but they take up from the environment. But so far, I haven't tied up how much they take from the environment with how much they grow. So basically, they create this gradient from the edge of the blob to the interior, because they are taking up some of the nutrients. And then next, what I do is that I I create an additional cell dictionary entry called uptake because I want to record how much my cells are taking from the environment. And then instead of using the traditional uptake function, I'm gonna use now this uptake inside cell total count option, which allows me to retrieve the number or how much the cell is taking from the field. And I'm passing that information to the dictionary entry. And I'm creating an additional field that would allow me to see how much my cells are visually, how much my cells are taking up. And I do it by going to CC3D Python, extra fields, automatic tracking, track scalar cell attribute. And notice that this has to be in the init function. And then I call that the field name uptake and the attribute name, which has to correspond to the attribute that I've created in the cell. I call it uptake. And when I ran that simulation, I can now display my cell field, my nutrient field that I see that it's decaying as the blob grows bigger. And that can I also visualize how much my cells are taking up from the environment. And I can see that the cells at the edge of the blob are taking more than the cells at the center of the blob. And now I can finally modify how the cell growth by how much my cells are taking from the environment. And I do it, this is the same equation we saw earlier. I do it by dividing by this, how much my cell is taking up by a reference number. Why the nutrient levels in the corner remain high? Because the setup for this particular simulation is that the medium is the one secreting, right? So at the edge of the boundary of the cell blob is with medium. Medium is secreting, so medium is always high, especially because I set it at a constant concentration. But as soon as the chemicals start diffusing from the medium, from the interface here inside the blob, then it starts being consumed by cells. So now I can modify how much the cell growth based on how much it's taking up. And I can play with how the diffusion coefficient affects how much my cell is growing. So if I make my diffusion coefficient smaller, that means that the molecule or the chemical field diffuses shorter into the blob. And I can see here that only the cells at the edge of the blob are taking up. So those are the only cells that are growing. And then as opposed to when my diffusion is bigger, that my chemical is able to diffuse further into the blob, and so cells are able to grow. Cells deeper into the blob are able to grow as well. So I still have the chemotaxis to introduce, but what I'll do is I'll stop here, and Gilberto is still gonna talk about how to implement chemotaxis later today.
Josh, I think
Yep.
chemotaxis is important enough that I would take the beginning of the next module and use it there.
Okay. Okay, I can certainly explain it in the next
So
module.
during the break, work with Giuliano to put that in at the beginning.
Okay, absolutely. And but before finishing here, I just want to make sure that I explain this well. So remember what I did is I have the chemical field being produced in the medium, and then the cells take up from that field. How much the chemical diffuses into the blood controls which of my cells grow and at what rate. If it diffuses over short distances, then only the cells at the edge of my blood are able to grow. If it diffuses deeper into the blood, then more and more cells are able to grow and divide. And I'm gonna stop here and then I'll continue with chemotaxis on the next module. So James, I'm handing back to you.
Okay, thank you, everybody. Please fill out the little questionnaire on this module, if you would. And we will start back at 12. It's 12 o'clock, so we'll start back at 12:15 for the next module. Thanks very much to everybody.